We are a cytogenomics lab, like a referral lab, and we do a lot of cytogenomic testing that includes conventional cytogenetics and molecular cytogenomics. So we do testing for a wide variety of clinical samples and sample types. Clinical samples means like prenatal samples, pediatric samples. We even do pre-implantation genetic screening, and a significant part of our work involves analysis of pregnancy loss. Most of the referrals that we get are basically to identify genomic imbalances, copy number variations, chromosomal abnormalities in the different types of samples that we receive, starting from prenatal, pre-implantation genetic screening like embryo biopsies, postnatal pediatric blood samples, and for adult patients who have reproductive difficulties. We haven't stretched the limit of what technologies we currently use, but we are hoping, NGS-based technologies, whether it's whole genome sequencing or whole exome sequencing, whether we can capture, genomic imbalances from, those technologies, with high sensitivity and specificity. Yeah, we, as you know, have been a long-time user of BioDiscovery's analytical and visualization tool, NxClinical, most recently. And the primary reason that we have continued to use it and hope to use it for some time in the future is, the robust nature of the software and the ease with which it allows us to analyze and review and identify, the types of genomic imbalances that we are trying to identify. And the ease with which this software, interacts and crosstalks with our laboratory information systems is a huge advantage. And additionally, NxClinical allows us to, analyze and look at, a wide variety of sample types that may not always be very optimally analyzed and visualized by other available tools. The microarray we use is a SNP-based array from Illumina that is a whole genome, provides whole genome coverage. So once the sample is processed and the microarray is run, the data is captured by NxClinical. And what NxClinical does is some processing and provides us a genome-wide view of all the data points across the genome for the SNPs. So that allows us to look at two very important elements that the data generates, the microarray SNP data. And that is copy number variation, whether there are large or small segmental deletions and duplications. And additionally allows us to look at the genotype data across the genome, because it's a whole genome SNP platform. So we capture, events that occur in the genome, copy number losses, copy number gains, or, regions of homozygosity or uniparental isodisomy. These are genomic events that have, immense clinical impact, often, responsible for a clinical phenotype or a disorder. And, the ability for us to, capture all these different elements of data from the microarray and visualize, process, and review and, make decisions for reporting to the patient through NxClinical makes, not only empowers us to provide answers to patients, but makes our, our life in the lab a lot easier, because the seamlessness of the interaction between the data processing by NxClinical and its ability to transmit and transfer data to our reporting system. The primary aim of, using these powerful tools in a cytogenomics lab is to identify structural variation or unbalanced structural variation in the genome that are responsible for a significant fraction of, clinical phenotypes. And these are not easily achievable by using, whole genome or whole exome sequencing, which is ideal for single nucleotide variation or small indels or small deletions. So when there are large unbalanced genomic events, tools such as NxClinical that allow us actually to visualize the data across all chromosomes, that's the entire genome, provides very high confident answers for genomic events that either are responsible or contribute to genomic phenotypes. We have been a very early and high volume user of NxClinical because of it not only does a very good analysis of the data that is generated from microarrays, but allows us actually to get a perfect visualization to add our own data sets that are used for comparative purposes. So it allows us to not only look at genomic events, at the microscopic level, exactly what genes are involved or which region of a chromosome is involved, but from the whole genome perspective, allows us to, provide answers about, specific events. And the ability for us to actually in real time, look at the data and make changes to the data, making calls non-relevant or relevant, or making additional calls or changing specific calls, the nature of specific calls is very important, and I think NxClinical allows us to do that, with extreme, ease. And that's very important for us because, sometimes, the calls may be, inaccurate or, partially inaccurate, and it is important for us to make those corrections in real time. And for have the data actually that we decide on is specific to be transmitted to our lab information system is very important. It's been a very productive, relationship, and we think that our interactions in terms of enhancing some of the things we are able to do and, co-develop with you or provide you suggestions and, reflect on changes that we would like to see in the future have been, extremely well accepted by, the BioDiscovery team. And we have, strong tools such as, NxClinical that allows us to, give, accurate answers as often as possible.